Hi, church family. This week is where we are restarting the apologetics series, and this is week number 5. The first 4 weeks we were discussing what apologetics was, and then going into how astronomy and the universe and how those things gave us some evidence as to whether or not there was a God. Um, this section is going to be talking more specifically about biology and how it points or doesn't point to intelligent design. So this week is titled DNA by Design. So to review, the last 4 weeks we were talking about whether we saw signs of the world showing intelligent design versus non-intelligent design. And in the last 4 weeks, we were talking about the universe. We learned that there was evidence pointing to the fact that it expanded from a single point origin, and that also the conditions that we find life in a stable universe today is 1 in 10 to the 43 chance of perfect conditions. And we also showed that we see recognizable patterns in the universe that not only show that it's improbable, but that it looks like there was some intelligent design. So the next section is talking about how biology shows evidence. I will not bias you into saying which way it points to, but yes, so biology, what will it show us about intelligent versus non-intelligent design? This is an excerpt from Charles Darwin's On the Origin of Species. So if you remember, Darwin is the father of evolution theory, where he believes that the world came into being not of intelligent processes, but of natural, undirected, natural selection. So I know this chart doesn't really make sense, but it's showing up here at the top that these are the species that we see today. These are the things that it came from. And down here is where it originally, more simple biology or biological things that it came from. So I'll show you a better pictorial graph that has more labels on it. So these are plants, protozoa, and animals that we see today up here in the upper section. This middle section is maybe things that they share in common. And then if you see, the more important thing is that if you see where it comes together, the root of this tree is this trunk is what's called a simple-celled organism. So Darwin talks about how it all started from a simple cell. They thought that this cell at this time was just like a soup of chemicals that came together and made this biological cell. You can see this is a picture of slime, but I couldn't really find any other picture to represent that. But basically, they thought this chemical soup of elements just kind of was, in the atmosphere, in the planet, and this coalition of elements came together eventually through just simple chance, and it made the first cell. And the first cell was thought to be just a simple homogeneous globule of plasma. Okay. So after Darwin, Oparin was a scientist that in the 1920s that he showed that yes, if we have these elements in the atmosphere and we zapped it with some kind of energy source, we would form we could form amino acids. These amino acids could therefore combine into these protein, these chains of amino acids, and those chains of amino acids could form a cell. These cells would multiply and, uh, or I'm sorry, this is primitive, primitive pre-cells, and then it would make the first cell. Okay, so that was his theory. And to test that theory, they actually did an experiment. They got a gaseous chamber and filled it with elements or molecules that they thought may be present in Earth's early atmosphere, like methane, ammonia, water, hydrogen. And they, what did they do? They added energy to it by boiling water. They, I don't know exactly, I can't remember this experiment, but they somehow added energy to the circuit, and then they found that organic molecules appeared after a few days. Okay, so at the time they were like, this is proof. This is proof of how life could have started without needing an intelligent designer. There are certain problems with this experiment. Later, they found that the gases that they put into this experiment were not really uh, likely to be the ones present at the beginning of a new planet. Um, the but the big discrepancy was the step from getting amino acids to a functioning protein. So see up here, we do get some amino acids being formed, but then to go from amino acids to a functioning protein, they just couldn't explain that step. So to better understand why they couldn't make that leap, we have to study what a protein is and how it's function in a cell. So just to let you know from the beginning, proteins are not very simple. I know you may not know what a protein is. You know that you've heard about it being, how much protein is in your diet or things that you eat. But proteins are actually um, like sequences of amino acids that have a very specific function. Though the dietary proteins are different than the proteins that we're talking. I mean, they are the same, but a simple protein is. We'll talk about that maybe in class. But in any case, the protein that us you would need to form a living organism um, is, you can see on this picture. This is a picture of the 20 amino acids. A protein from a single cell um, is on average about 240 to 350 amino acids long. Okay, so they are very specific. Not only do they are they specific in the sequence, like these are all very unique, different types of amino acids. So these 240 amino acids are in a very lettered sequence, okay? Not only are they specific in a sequence, they're specific in the way they fold upon each other. So you might have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and then it folds, 1, 2, 3, 4, 5, 6, 7, 8, and then it folds. So where they fold is also very specific to its function. And then finally, the way this folded sequence of amino acids uh, are folded in a three-dimensional way, that is also very specific to how each protein functions. So without this specificity, they do not function in the cell. They know this because they've taken one little protein out of a sequence of a protein, and then it doesn't work. It doesn't do its job like it used to. Or if they take away the folded, they change the way it's folded, then it doesn't work either. Okay, so that's what it means when it says it's very specific. And to go even further, they found that these proteins, not only are they specific in the way that they are made, they are very specific in its genetic code. of how it's made. So the video I will show you will kind of explain how proteins are made from DNA. DNA are not proteins. They are kind of like computer code, and it tells you which amino acids go in which line, in what sequence, and how they are put together. So, I mean, I'm telling you all this within a span of 5 minutes, but these are all very revolutionary things that they found in biology. So that not only did they find that these proteins are very specifically made and very specific in their length and what was on them, but they found that there was a computer, almost like a metaphorical computer, that told how the proteins were to be made in each cell. Okay, so here's a video that kind of explains it better.
That chemicals, called bases along the spine of the DNA molecule, function as alphabetic characters in a written language, or digital characters in machine code. This short animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation ensues. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cells by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. Sequential arrangement of the amino acid determines the type of protein constructed. When the construction of the chain is complete, it is often transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell. We have cells.
Okay, so that kind of gives you an overview of how complex protein synthesis is, from its decoding from the DNA to how it's made, how it's folded, and then transported out of the cell. I mean, it's really, really amazing. Even the most simplest cell, how it functions. So initially, if you see this picture that we showed you in the beginning, these elements turning into slime, we can X that out because this simple slime just didn't exist. The more they were able to study the simple cell, they found it to be more like this. Okay, it had I mean, this is an animal cell, but this, has all these elements, proteins, nucleus with DNA, performing all these functions. It was much more complex. This step seemed a much bigger leap than they had originally thought. So, to review, what steps would have had to what steps would have had to be made from going from gaseous elements to the formation of a cell, first you would have to have the right gaseous elements. Those would have to make the 20 different naturally occurring amino acids that we see today. Those amino acids are in a particular sequence, the smallest being 140 long and the longest being tens of thousands long. These specific amino acids are linked together and called proteins, and then those proteins perform specific functions so that the cell can live. Okay, um, these proteins in the cell are organized into organelles, sectioned off into the nucleus, the nucleolus, and the cell body. Um, and then, of course, you know, these cells are eventually organized into higher functioning beings like animals, birds, and intelligent human beings. I'm sorry, intelligent human beings, or not so intelligent human beings. Okay. So just to review what is being said, the materialist view, or the naturalist worldview, says that there is no intelligent design. Everything that's here today is a process of undirected natural processes, or everything evolved as a process of chance and natural selection. Okay? The theistic view is that there was intelligent design and that proteins, cells, and life as we know it came about through intentional, intelligent design. Okay, so those are the two worldviews that we're discussing. So the evidence that we have looked at so far, the critical point of contention is the step from the gaseous elements to a simple protein. Proteins that the closer we look at them are very complex. They perform specific functions of the cell. They not only are exact in their sequence, if you get one of those amino acids wrong, they don't work anymore. Not only is the sequence critical, the folding of the sequence is critical. And then the three-dimensional folding of the amino acid sequence is critical to its function. Okay, and not only that, proteins, the way they're sequenced is encoded by this crazy computer that's in each cell called DNA, and they direct exactly how to make these proteins. Okay, and the startling fact of this is there are no junk proteins. There's no junk DNA. Okay, so I'm going to repeat that. There doesn't seem to be any junk DNA or junk proteins. So if we had a process of natural selection where we just made all these weird proteins and then the fittest proteins would be naturally selected, you would see some proteins that were junk that didn't really need to be there, but that just happened out of natural sequence, or that just happened out of natural chance. But we don't see that in life. Every protein serves a specific critical function of the cell. Not only that, the miles and miles of DNA in each cell all are critical to the functioning of the cell. So there is, at first they thought that there was junk DNA or DNA that didn't really seem to be of any use, that it just happened to be there. They're finding more and more ways that every single sequence of a DNA is critical. Okay, so this is so interesting, and can't wait to talk to you about it this Saturday. And bring all your questions, and looking forward to discussing it with you.